The first up, of course, is our patron, Professor Mary Reilly. Needs no introduction, an absolute authority on CMT in the UK, and delighted, as always, to have her on board. I certainly don't get to spend enough time picking her brains on CMT and the amazing work that her and her team do. But we are absolutely delighted to have her again on board, sharing in her own time and what's been happening in the CMT world here over the last 12 months. So Mary, I'll pass on to you with far more important words than I could possibly deliver.
Thank you very much, Paul, for that very kind introduction. I'm absolutely delighted to be here again this year, and we're all getting used to the virtual environment, although it would have been nice to see you in person. I wonder, can someone confirm if they can see my slides before I start?
Yes, slides are on screen.
And full screen. Perfect, thank you. So I'm going to give a talk about the opportunities and challenges in CMT therapy development. As when patients come to clinic, mostly patients are interested in what's happening about treatment. I mean, it's almost the first question people ask after getting a diagnosis. And I'd like to just show my disclosures, but none affect this talk. So I'm going to give a very brief introduction, describe my view of therapeutic approaches, and then say what I think is most promising and is likely to be seen in the next 5 years. So I'm talking today about Charcot-Marie-Tooth disease, and I'm just going to define what I mean by that in terms of what I'm talking about, because there are literally hundreds of diseases where neuropathy is part of them, which are inherited. But when we talk about Charcot-Marie-Tooth disease, we're really talking about a family of diseases. So Charcot-Marie-Tooth disease is the original inherited neuropathy described with motor and sensory involvement. It's related genetically to hereditary neuropathy with liability to pressure palsy. If you have pure motor or almost pure motor involvement, it's called hereditary motor neuropathy. And if you have pure sensory or almost pure sensory, it's called hereditary sensory neuropathy. But when we talk about CMT, we mean this family of diseases. And the reason we mean this family is that there's over 100 causative genes described. But if you look at these genes, and this is the dominant group, and this is the recessive group. But in each group, if you look at the actual diseases, you have CMT1, 2, hereditary motor neuropathy, hereditary sensory neuropathy, there is an overlap in the diseases seen with the genes, which is why we now talk about them as a family of diseases. So for the rest of the talk, when I refer to CMT, I'm referring to this family of diseases. Now, when we talk about therapeutic approaches, now what I'm confining myself today is pharmaceutical therapeutics. So clearly, there's a huge amount going on therapeutically in terms of orthopedic management, orthosis management, therapy management, pain management, which I won't address today. And what do I think we need to think about with therapies? So to talk about therapies, for a patient to ever get a therapy, they need a diagnosis. We need candidate therapies. We need clinical trials of those therapies. And to do the clinical trials, we need responsive outcome measures. So all of those steps have to be gone through to have a therapy for CMT. So how good are we at the diagnosis? So in the UK currently, we are reasonably good at diagnosis. So we published this paper last year, which really compared UK practice, which is where we do diagnosis through the NHS in accredited hospital-based laboratories. And we compared that to US practice, which is my colleague Mike Shai runs a consortium in the US. And their diagnosis is done through commercial companies. But really, it should be equivalent in terms of the two groups. And we did find we had an equivalent diagnostic rate, and it was about 73%. And there was a certain group of genes which we both identified as being the most common causes of CMT. And in my own practice in the UK now, we have about a 77% diagnostic rate in clinic. So if you go to clinic, there's about a 77% chance you get a diagnosis, and we still don't know just over 20% of the genes. And as you'll see here, we're much better at CMT1 than we are at CMT2. And I just wanted to mention what's changed in the UK. Over the last few years, the UK has moved to a UK-wide diagnostic platform. And in England, this is called the R78 Neuropathy Panel. So this means that wherever you are in the UK, if you have a panel of tests done for CMT. It's exactly the same panel you'd have done in any lab, and there's accredited laboratories doing this. And this is now done by a technique called whole genome sequencing, which means when you consent for your genetic testing, you can also consent for that to be used for research purposes if you're one of those 23% who don't have a diagnostic gene. So this has been a really good improvement in diagnosis in the UK. The fact that wherever you're seen, you get the same panel of genes done. And we and others that are experts in this input to the panel, we would say when a new gene needs to be added onto the panel. And this new platform has been enabled by the 100,000 Genome Project in the UK. So I think this has changed. So it's probably worth remembering R78 because people are beginning to use this term for the neuropathy panel now. And I think that's very helpful. The reason it's worth remembering you can also consent for research is if you look at a typical gene, and this is what a gene looks like when we show it diagrammatically. These dark grey parts of the gene are called the exons. And that's the part of the gene that's translated into a protein. And if you think about your whole genetic material, that all the exons of all of our genes, and we have 20,000 genes, only make up 2% of our genetic material. So when we look at a panel of genes, we're only looking at the exons of these genes. And to date, most of the abnormalities found that cause genetic diseases are in these exons. Not because there are none elsewhere, but that's because what we look at. But we know that there's a huge amount of genetic material, as you can see here, that accounts for the other 98%. And we now know from whole genome sequencing that what we find in this 98% are elements that influence these 20,000 genes. So you might have a gene that looks perfectly normal when we look at it, but it's something here that influences how this gene works in you that causes the disease. So that is why the 23% that are undiagnosed are probably problems within the genome. And even people that have a diagnosis, if you think about your family, one person might have it more severely than the other. It is likely to be problems in the whole genome that affect that. So the ability that we have in the UK for everyone to get a whole genome sequence so that we can not only give a diagnosis, but also in the research environment begin to look at these other modifiers is to me a huge development in the last few years. So one of the things I'm interested in is research to close this diagnostic gap, that 23% missing. And I won't go into the details of that today because that is not what the talk is about, but really that's what whole genome sequencing is allowing us to do. So of course, when you have a diagnosis, the next step is developing candidate therapies and doing clinical trials. And I will deal with this next. So when I think of therapies, I think of what are the targets. So we are either developing therapies against a single gene, for instance, PMP22 and CMT1A, or targets against multiple genes in a common pathway. So they are the targets we think about. And the kind of therapies we think about are either generic gene therapies, and I'll describe all of these, therapies that target the pathogenesis of the disease, or pathway therapies. And the generic gene therapies are the area where there has been massive development in the last 5 or 6 years. And there's reasons there's been massive development is the technology is applicable to all hereditary diseases. So pharma companies are interested. So if you develop a technology that you can modify to use any inherited disease, clearly that's attractive. And that has revolutionized rare diseases. And if we think of the generic genetic therapies, these are therapies that allow us to go back to our DNA and RNA in individual cells and change it. And I, for one, would never have believed this possible 20 years ago. So that is what I mean by generic genetic therapies. And in the next slide in red are all of the ones that are currently in patients. So therapies like antisense and small interfering RNA are therapies that change what happens at the mRNA level before it becomes a protein. And these are licensed for TTR amyloid, for spinal muscular atrophy and Duchenne's muscular dystrophy already. Going one step back to your actual DNA, in genes where the gene just doesn't work, you can replace the gene by packaging it in a viral vector, a bit like they do with some of the coronavirus vaccines, and give gene therapy that way. And again, in SMA, this is currently in practice. And then there are new technologies that allow you to go into the cell and edit the DNA. In other words, manipulate it and change it. And one of these is CRISPR-Cas9. And although these are not licensed as therapies, the very first humans to be given these was for an inherited neuropathy called TTR amyloidosis in 2021. So all of these in red are in patients, either as clinical therapies, these three, or as a therapy in trial. Now, the more traditional way of looking at the treatment of genetic diseases is to target the pathogenesis of the disease, for instance, PMP22. So that is where we find a gene, we work out how the gene affects the function of the normal protein, and then we target that function. We try to correct that function. So if you look at CMT1A, we know that it's a dosage problem. If you have 2 copies of the gene, you're normal. If you have 3 copies, you have CMT1A. And indeed, if you have 1 copy, you have HMPP. So in multiple models, in animal models and in humans, we know it's a dosage problem. So the therapies, many of them that are being developed for CMT1A, are therapies that will manipulate the dosage and reduce the amount of PMP22 expression. So that's what we call a pathogenesis therapy. And the third kind of therapies are the ones that are really attractive, but a little bit further from development. And these are, let's take a full pathway that has multiple genes in it, and let's try to treat the pathway. So if you go to your doctor with high blood pressure, they treat the high blood pressure despite the fact that there'll be multiple different causes. So that's a pathway they're treating. And of course, this would be fantastic for genetic diseases, because if you have one of those rare CMTs that affects 10 families in the world, if a pathway is being treated, you're more likely to get a therapy developed quicker. And of course, this applies to other diseases that might affect the same pathways. There might be acquired diseases. For instance, many chemotherapies used for cancers cause neuropathy. And if we found a pathway of in the nerve that treatment would work for genetic diseases and also prevent the neuropathy of chemotherapy, that would be huge. And in CMT, we've identified multiple pathways, and all these pathways in red have multiple genes in them. And one of them is axonal transport, because we know that the axons in humans are long. If you're a 6-foot man, you probably have an axon from your lower spinal cord to your toe that's 1 meter long. So there's a huge amount of transport of nutrients up and down, and multiple genes involved in that. And there are, at the preclinical level, that means not yet in humans, there are multiple products being developed to try to treat these pathways. That is complex, but I do think it will deliver in the long term therapies that can cross multiple forms of CMT. So for the rest of the talk this morning, when I talk about any therapy development, I will constantly refer back to what is the target and what type of therapy it is. Now, what is happening? So if you want to know what's happening in CMT or in any disease, we go onto a website called clinicaltrials.gov, and that tells you how many trials are happening. Now, I was very excited when I looked it up for this talk, because it says there are 455 trials happening in CMT. Now, when you break that down to trials that are actively recruiting, that means they're currently actually engaging in patients. There were only 82. And unfortunately, when you actually break that down to which one is a therapeutic trial, there's only one. So the other trials might be a trial of an exercise or a trial of an orthosis or a trial of something else. It doesn't mean it's not important. But at the moment, registered, there is only one trial. Now, that doesn't mean there aren't other trials planned or happening, but most people would register a trial if they were doing them. Most of us have to register a trial to get ethical approval. So a lot of what I'll say today is in the preclinical literature. That means it hasn't yet got to patient trials. So I am going to talk today about the most common forms of CMT and update you on what's happening, and then mention 2 rare forms that are going to trial in 2022, just so you're up to date. So despite 100 genes or more being described for CMT, CMT1A is still the commonest. And this international consortium study really throughout the UK, US, Northern Europe, Australia in 2015, 61% of patients had CMT1A. So it is the commonest in most countries. And as I said earlier, we looked at the other common causes, and they are JGB1, mitofusine, MP0, and SH3TC2. So that's CMT1B, CMT4C, CMT2A, and CMTX1. So these are by far the commonest, and I will talk about these today and then mention the two rarer ones which trials are about to start on. So as I said, CMT1A is due to an extra copy of the protein PMP22. You basically have too much of the protein. And we know that the dose of this protein is critical for myelin, and that if you don't have the right dose, you get a neuropathy. We also know CMT1A is a slowly progressive but very disabling lifelong condition. And that is very important to keep in mind, because many of the therapies that are now in practice, and some of my patients get very frustrated why CMT hasn't got therapies. But that's because SMA, Duchenne's are fatal diseases if not treated. So when you're developing a therapy, you're allowed to take it to clinical trial much more quickly if it's a fatal disease than a chronic disabling one. Not that people don't think CMT is important, but safety is paramount. You have to make sure that you have a drug that will not affect life in any way if you're going to live a whole lifespan. So most of the therapies that are being developed try to reduce the amount of PMP22. And in this very nice review by Parison last year, it looked at all of the different ways that people are trying to treat CMT1A. And I'm going to stick to the overexpression ones, but there are other ways, most of them that are not near clinical trials and may or may not get to clinical trials. But certainly reducing the expression is the most important one. So the target here is PMP22, and they're targeting the pathogenesis of the disease, that is overexpression of PMP22. And the first one that came to clinical trial was vitamin C, which was shown in a mouse model, and there was a rationale for this, that if you treated with vitamin C, the mouse did improve. But we know, and many people in the UK were involved in the UK-Italian study, but multiple studies have shown that CMT does not improve patients with CMT1A. And what we learned from the trial, this is the UK-Italian trial, this is a study over 2 years, and the blue is patients who are on CMT, on vitamin C, and the red is patients who are on placebo, that is not an active drug. And this is our outcome measure. So what we learned is, we actually didn't know how to measure progression in CMT over 2 years. So many of you will have shared the frustration of going to a clinic and saying, I feel a little bit worse. The doctors examine you and say, everything is stable. That's our fault. So our clinical measures, we do pick up change over 4 or 5 years, but most of them do not pick up change over the year or two you need in a clinical trial. So that has really stimulated us doing a lot of work before going to further trials. Now the second drug that was tried to reduce PMP2 expression was a progesterone antagonist, so it again works in an animal model. But progesterone is a female hormone, and as you can imagine, there are safety concerns about using this. And although there is a newer version that has gone to clinical trial in France, it's never been completed, the results have not been published, and it's not recruiting. So we can only assume that this did not work. The third drug I wanted to mention is the drug that is undergoing a clinical trial called Farnext. And it's called PXT3003. And this was a different way of developing a drug. So what this company did is they had a hypothesis that they would look using computer modeling to see what kind of drugs that are already out there might affect the expression of PMP22. So they modeled this by looking at the pathway of PMP22, and they picked 3 compounds: an opioid antagonist called naltrexone, a GABA receptor agonist called baclofen, and sorbitol, which affects a muscarinic receptor. So from their computer models, they thought that this combination would affect the expression of PMP22. They showed in an animal model that there appeared to be improvement in the model, and that the PMP22 expression reduced when they used this. And they then moved to a safety phase 2 study and showed it was safe, which was not a surprise because the 3 drugs are used in practice and they were using very low doses. And there was a hint in this trial that there may have been an improvement. So they moved to a phase 3 trial, which is a trial where they have multiple patients throughout the world. And in the higher dose of the drug, they did show an improvement in 2 outcome measures, a 10-meter time walk and the overall neuropathy limitation scale. Now, there are some issues with these outcome measures. Now, first of all, this is no criticism at all of the outcome measures they picked, because the licensing authorities that let drugs be developed, like the FDA and EMA, they influence what outcome measures are used. So they were involved in the deciding to use these outcome measures, but we had already shown in studies that they don't change in a couple of years in patients. So if the drugs stop progression, we don't think they would pick it up. Clearly, if drugs cause a dramatic improvement, they still would pick it up. But it goes back to this question is, how should we measure it? And also, it's our responsibility, if we think we've better measures, to work with the FDA and EMA to make sure that companies are allowed to use them. Now, unfortunately, in that trial, even though they did show some improvement, there was an issue, just a pharmacological issue with their high dose, in that it crystallized and they had to use twice the low dose product rather than a high dose. And when it came to lysosine, my understanding is that they were unhappy and they suggested they would repeat the trial. So the phase 3 trial is now ongoing, and my understanding it is fully recruited and started in 2021. And they have picked the same 2 outcome measures that they had used in the previous trial. So when I said there's a trial ongoing, this is the trial that is now ongoing in CMT1A. Now, the very exciting development in CM1A are these gene therapies. Now, this again is looking at the target of PMP22, looking at the pathogenesis, but using a gene therapy to treat the pathogenesis. And the 2 that are of particular interest in CMT1A are antisense and small interfering RNA. And what I mean by that is these compounds, these genetic therapies, are used in the following way. So in all of our cells in our body, we have DNA, and DNA is the code for all of our genes to become proteins. So when the DNA makes a protein, it gets transcribed into what's called messenger RNA, and the messenger RNA then gets translated into a protein. So what the antisense and the silencing RNA can do is silence the messenger RNA or reduce the amount that's translated. So we can act at the cell level to reduce the expression, and that's exactly what you would want in CMT1A. And this has been used incredibly successfully in another inherited neuropathy, TTR amyloidosis. And in this case, we wanted to completely turn off the production of the TTR because you don't need the protein in the liver so that it wouldn't form the amyloid that affects the nerves. And the 2 gene silencing therapies that are now licensed and patients are getting shut down this production by 80 to 90%. So they're incredibly effective. So the proof of principle is these could be very successful for neuropathy. And in an animal model, and there'd be multiple models, and I'm only mentioning one because I think it's the best paper, where they looked at both a mouse model and a rat model and looked at what I think are a really good set of outcomes, because you have to be very careful that you look at outcomes in animals that are relevant for humans, because mice are not men. So, but it's really important we pick the right outcomes. And multiple trials in multiple diseases have failed. But they looked at how the animals behave. They did nerve conduction studies, they did pathological studies, and they did PM2 expression. So I think this is really exciting. And this is at the moment in preclinical development in multiple different ways. The difficulty is they need to get the dosage right. You want to reduce the expression of PMP22 by 1/3 so that you have equivalent of 2 copies, but you don't want to go too low or you get HMPP. And I think this will come to trial in the next few years. And this technique has been used for multiple other diseases, but as I said, the safety has to be absolutely got right for PMP22, and also getting it into the Schwann cells, so getting it into the nerves. It's no point, if the liver doesn't produce it, we need to get it into the nerve. And that has been overcome, certainly in animal models, and there's been some incredibly good work to overcome that in humans. Now, moving on, the second form of CMT that we normally talk about is CMT1B due to myelin protein 0 mutations, and they can also cause an axonal neuropathy and a recessive neuropathy. And they account for about 5% of CMT in the UK. Now, in this case, the therapies that are being developed, they're looking at the single gene and they're looking at pathogenesis. And this is a little bit more complex because we do see different phenotypes of these mutations. We see patients who present very early on with a very severe neuropathy, which is demyelinating, and very severe sensory involvement with pain and unsteadiness. And then we see a form that presents much later with a milder neuropathy, and which there is mainly axonal. So this gene really causes CMT1 and CMT2. But we do know that many of the mutations in the gene affect what's called the unfolded protein response. So if your protein, when it's made, is not folded properly, it builds up in the endoplasmic reticulum, and then the body deals with this by turning on this pathway, the unfolded protein response, to get rid of these unfolded proteins. But eventually the pathway becomes overwhelmed, and these unfolded proteins can actually begin to kill cells. So this is very important in CMT. In CMT1B, multiple mutations have been shown to affect this unfolded protein response. So there are two interesting drugs. One was curcumin, which was used in an animal model, but it's not suitable for humans. And a drug has been developed, Cephin-1, by a company, Inflectus, which does modulate part of this pathway. And certainly in animal models of 1B and 1A, it does seem to improve the neuropathy. So this is currently still in preclinical development, but it's an interesting development because we do know this unfolded protein response is also involved in other forms of CMT and other inherited diseases, but that's at the preclinical level. The next two forms of CMT I'm going to talk about is JGB1 and SH3TC2. So I'm now moving to forms of CMT where the problem is when you have a mutation in a gene, the protein just stops working. So in a way, that's easier, because if the protein stops working, you can replace it. Whereas if the protein is toxic to other proteins, then it's more difficult. So this is done by viral vector-mediated gene therapy. So if the protein isn't working, you actually give the gene, and the gene is the DNA, you wrap it in a viral vector, as I said, like you do for a coronavirus vaccine, and you get this into the cell where it can be incorporated into the gene. So if you look at it here, you have a protective gene, say, it gets wrapped in a viral vector, it's get delivered to the cell, and in this case it will be the Schwann cells, or in other cases it will be the axons, and then the protective viral vector allows the gene to escape, and the gene goes into the DNA. Now, the virus is inactive, so it's not, that's what people always worry about, it's not a virus inactive, but this is very promising, and again in SMA, this is currently one that has been licensed. Now, for CMTX1, this I would say is the target, but again, we're targeting the pathogenesis of the disease with the generic gene therapy. So that's very useful. And as we know in X-linked CMT, due to JGB1 mutations, male are more severely affected than females because they have only one X chromosome, where females have two. And in males, because it's loss of function, most males at 20 or at 30 or at 40 are very similarly affected to other males. So compared to other diseases like of CMT, other types, we see more uniformity of the effect in males. In females, it's very different because some cells express the X that's involved and some cells don't. Now, we know from work I've done that about 10% of patients who have a problem with the JGB1 mutation, it's actually a non-coding problem. I mentioned that to you earlier. This is something we'd have found by whole genome sequencing. And this tells us that many other genes will be the same. But we know that these non-coding mutations also cause loss of function. So the leader in the field, in this field, is Cleopas Cleapa in Cyprus. And he has developed a therapy which does work in an animal model. So that is, if you give it by a lumbar puncture and you give it, they will improve the neuropathy. We've worked with him to show that this biomarker we've developed for humans, neurofilament light chain, is responsive. So that's very encouraging. The second is a similar mechanism. SH3TC2 is the commonest cause of autosomal recessive CMT in the UK. And again, we're talking about the pathogenesis generic gene therapy for a single gene. And this is a very early onset, severe form of CMT, which is demyelinating, but with very early scoliosis and large fiber sensory loss. So this one's, and again, Cleopas is the person who's led on this, again with our biomarker, and shown that you can, again by intrathecal injection, by a lumbar puncture, replace the gene. Now, this is very exciting, but in the last year, there has been some safety concerns in another muscle disease using what's called AAV9, which is the viral vector. So there is some work to do on making sure the viral vectors are safety. And again, in CMT, these are two lifelong conditions. that will not kill patients until they die from natural causes. So safety is absolutely critical. So I think this is a little further off from going into patients than we would have wanted, because there is still some safety work to do. Now, the final common form of CMT I'm going to talk about is mitofusion 2. This causes CMT2, the axonal form of CMT2, and accounts for 20%, so it's by far the commonest cause. So the therapies, multiple therapies are being developed for this, and they mainly look at the pathogenesis of the disease, and that is obviously a single gene. So what does mitofusion do? Well, mitofusion is a nuclear mitochondrial gene, so it causes these mitochondria, which are like these energy batteries we have in every cell in our body. It causes them to fuse together, and then another CMT gene, GDAP, causes them to what's called fission, to split apart. A mitochondria need to keep fusing and splitting to keep working. And mitochondria drive the energy that goes up and down axons, and I mentioned to you how important these long axons are to humans. Now, it's been known for some years that there is another form of mitofusion called mitofusion 1, which really does exactly the same thing as mitofusion 2. But this is poorly expressed in nerves. It is found in other tissues, which is why maybe that we don't see other tissues involved so much with mitofusion 2 mutations, that mainly it's neuropathy. But people have wondered whether if you increase the expression of mitofusion 1 in nerves, could it compensate for the problem in mitofusion 2? And the earliest work done on this was in 2012 by Bob Balow, who showed in a culture that this was true. You could rescue the axonal degeneration. And he's moved on to show in animal models that you can do this. Now, there are multiple companies working on this now, working on different ways of manipulating the system. So you downregulate mitofusion 2 and upregulate mitofusion 1. But this is pretty preclinical. I would be surprised if there's a clinical trial in this in the next 2 or 3 years. But there will be. Maybe it's 4 or 5 years down the line. But, we can get it wrong. If somebody finds something quicker, it may be developed. And I'll tell you how that can happen in a few minutes. So really, that's covering the commonest causes of CMT. And if you think that I said we make a diagnosis in 73% of people with CMT, these 4 genes account for 91% of those 73%. So if we can develop therapies for this group, we've covered most of CMT. But I'm now going to move to 2 types of CMT, which we will be starting clinical trials in the UK in 2022. Now, the first one is very close to my heart because it's basically very much a UK-focused disease. It's called hereditary sensory neuropathy, which is a terrible name because it's severe motor involvement. And it's a disease that starts usually about age 13, 14. It starts as a sensory neuropathy, but develops a motor component. So patients start with the complications of not being able to feel. So usually at 13, 14, patients find they've cut their foot or they've done something they didn't feel. And these are the complications when you cannot feel. And I really always want to make the point that loss of sensation is as important as loss of muscles. So you develop cuts, you develop blisters, you can have amputations because you had an infection that went into your bone, and you can have what's called Charcot joints. But these patients with time develop severe motor involvement, more severe than many patients with CMT. And you can look here, very severe wasting. Look at the hands here in these patients. So it is a very severe form. Now, I estimate there's 250 to 300 patients in the world with this. But there's over 100 in the UK because there's a founder effect, which we published about in 2006. And the original family in the UK was described by Denny Brown in 1902. And as you can see, these are not small families. There's probably many other patients in the UK not diagnosed. But this is a disease which I have a particular interest in. I follow up about 70 patients. And we have worked out what causes this disease. So there is an enzyme called serine transferase that condenses these 2 chemicals to form complex phingolipids. And in dominant diseases like this, that is a disease where a patient has a 50-50 chance of passing it on to his daughter or son, we know that enzymes like this, if you only have a problem in one copy of the enzyme, that doesn't cause the disease. So it's not because of loss of 50% function. What a very clever chemist called Thorsten Horneman showed by looking at our patients is that what happens is when you have a mutation in the 2 genes that make up serine transferase, that instead of this happening, which is the normal reaction, this happens. So the palmitoyl condenses alanine and glycine to form these complex sphingolipids called DSPs, which are neurotoxic, that is, toxic to nerves. And he postulated and showed in an animal model, if you gave the animal serine, it pushes the normal reaction. The DSP levels fall and the animals get better. Now, the problem with it wasn't a great animal model because it had very mild neuropathy. That is one of the problems. So two things happened after that. In the US, they did a clinical trial in humans, but it's a very rare disease and they had only 18 patients. And their outcome measure they used wasn't sensitive, so they didn't have a positive clinical trial. And what we mean by that is what they said was a primary outcome measure didn't work. But they did have some promising results. And because of those, we thought we should do things differently in the UK. And I won't dwell on what we did, but we've spent years developing an IPS model. That is where we took skin from patients, grew the skin, used cells to go back to what we call pluripotent stem cells, and then made neurons. And using those neurons, we worked out exactly the cause of the neuropathy and showed that serine works. So we showed that given serine increases, the patients have, or the cells become better. And if we give alanine and glycine, which is the opposite of serine, they get worse. Now, the second thing we needed to do is think about, this is a really rare disease, and many forms of CMT are rare. So how are we going to do a trial and get an answer? We know from the vitamin C trials that even when we have a common form of CMT, it's difficult. So I've spent the last few years working with many patients throughout the UK to do a natural history study. And I was very grateful to the MRC who funded this. And this study, we looked in detail at all of the outcome measures that we would expect to change over a year. And we found that a protocol we developed here in Queen Square to look at fat in muscle was the most sensitive. So when your nerve to your muscles starts to not work properly, the muscle becomes thinner and develops fat. So this is a normal muscle. This is a muscle here, or here, or here, which has been replaced by fat in the muscle because the nerve has died or is dying. We can measure this fat really accurately in the thigh, in the calf, and in the foot. And by doing that, we've been able to show that we can show change in 1 year in patients. And we've done that now in 5 different types of CMT, including this one. And this allowed us to develop a protocol for a clinical trial, which allows us to use every single patient in the UK, because we can use a different outcome measure depending on how severe the fat infiltration is in the individual patients. And I've called this the SENSE trial, and I'm very grateful for the CMTA who funded this trial, and we're starting in June 2022. And for anyone that's listening that does have this type of CMT, please get in contact with us, because we are gradually contacting all patients, and we will do an information day to patients virtually beforehand. And the final kind of CMT I'm going to talk about really is a very interesting story. So basically, this is Andrea Cortesia, and Andrea Cortesia was a clinical postdoctoral fellow working with me, and whom I recruited into what's called INC fellowship that I run. And this is a fellowship where people are allowed to do different things. And Andrea spent a couple of years with me and then 6 months with Stefan Zuckner in Miami. And by having a really good fellow that was able to work between laboratories, we worked together to find this gene. And you will see, I put this up on purpose. If we're going to find genes now, there's always international studies. We need everyone working together. But this was a really lucky gene to find, because it was a gene that an awful lot was known about. And it has allowed the gene to be found in 2022 and the clinical trial to be started in 2022. So 2 years from finding a gene to clinical trial, again, an amazing development. And the reason is because this gene is called SORD, sorbitol dehydrogenase. So it's a recessive disease. So if you have 2 abnormal copies of the gene, you have basically, this doesn't work. And if this doesn't work, the sorbitol increases. And we know from the diabetic literature that sorbitol is very important at causing neuropathy. So that's what we knew already. And the neuropathy, patients either are told they have CMT2 or HMN. So it's a motor predominant neuropathy. So these are some of my patients. So a lot of wasting in the legs, worse distally as you would expect, and less in the hands. And this is one of the patients. And in my practice, it accounts for between 7 and 9% of undiagnosed patients with distal HMN or CMT2. And since we published the paper in 2020, other papers find it in 3 to 6%. So it's not a common cause of CMT, but it's a common cause of the unexplained, if we put it that way. Now, what was really exciting, because we knew about the pathway, what we knew is, as I said, with this gene, sorbitol doesn't work, the dehydrogenase. The sorbitol goes up, and we know it causes the neuropathy. But because this has been studied in diabetes for about 20, 30 years, we knew there were a group of drugs that have been developed called aldose reductase inhibitors. So if you stop this, it stops glucose going to sorbitol, and it will prevent the sorbitol increasing. And basically, there are 2 drugs available. Now, they're not licensed in the UK or in the US because they actually have a good safety profile, but not brilliant. So there are some safety issues, there are side effects. But we were able to use them in our Miami group to look at a fly model of this neuropathy, and it improved the model. And then we were very lucky because a company called Applied Therapeutics had already developed a new safer aldose reductase inhibitor for another condition. And they tried it in our fly model and it worked. And they've now done a safety study in patients to show that it's safe, and it is. And they have now just announced the first trial, and we're working with them internationally to do this trial, including using our MRI outcome measure. And this trial is starting this month, and it will be an international trial, and we will be a UK centre for the trial. So I think this is serendipitous. It was because we found a rare gene in a pathway that already was known about and a company was working in. So because it was being worked in diabetes, it allowed, even with COVID for the last 2 years, to go from finding the gene to clinical trial in 2 years. Now, I'm very, very conservative when patients ask me, if you find my gene, how long will it take to develop something to clinical trial? And if you ask about CMT1A, it took about 20 years. So this is an extraordinary story, but this is happening more now because of technological advances. So what's the elephant in the room? Well, the elephant in the room is the outcome measures. How do we do clinical trials? So many of you, as I said, will know that you definitely think your CMT progresses. And I often find patients say, did it change in the last year? And they say, not sure, maybe a little. Did it change in the last 5 years? Absolutely, definitely. So I think we know it's progressive. We know there are times in your life, like during puberty and the teenage years, that it's more progressive. And often people, when they're older, feel it's more progressive. But that may be because they've less to lose. So if you have 100% normal axons and you lose 10%, you're probably still normal. If you have 50% and you lose 10%, it'll be a severe effect. So the less you have, the more a little loss causes a problem. So as I said, our clinical outcome measures are not that responsive. So what are we doing about that? Well, how do we measure CMT progression? So we use clinical examination and the history. How are you? We ask the patients what's changed. We examine them. We use nerve conduction studies, which are very good at telling us the severity, but have not proved to be very good at following people up for short times, like 1 to 2 years in a clinical trial. And we use numerous clinical scales that combine clinical and neurophysiology. Again, really good at showing severity and actually really good at showing progression over 3, 4, 5 years, but not so good at showing it over 1 or 2 years. So my group has been involved, as I've said earlier, in developing the MRI, muscle fat infiltration. My group and others have developed blood biomarkers, and other groups have developed skin biopsies like the PMP22. So this is, as I said, the fat infiltration. This is someone with very mild CMT, practically no fat in the muscles. This is someone slightly older with more severe CMT. You're beginning to see fat here. And this is someone older again with much more severe CMT. And really, once you get to this stage, you will not reverse it. So I think when we're thinking about therapies for CMT, my feeling is we're actually aiming at therapies at the moment that will stop progression. Now, we hope we might improve. And if we get someone at this stage, we might improve. But generally, when it's more advanced, we're looking at stopping progression. So any therapy that gives improvement will be a bonus. But at the moment, we're not so good at making axons regrow. But the younger you are, the better your system is at doing that. And as I said, we've shown this to be responsive in CMT1A, and we've shown it to be responsive now in HSN1, and that's published. An unpublished work that today I'm really announcing it, and it will be presented at the PNS next week in Miami, is we've now shown it to be responsive over 12 months in CMT-X1, 1B, and 2A. And this is in work which was funded by, for me, by the MDA in the US, and I'm really grateful. And as I said, we and others have developed some plasma biomarkers, so you take a blood test and we can show that this is responsive in the neuropathy in animal models, and that certainly it correlates with severity in humans, although we've yet to see will it be responsive in trials because we haven't done the trials. So to finish, what do I think will happen in the next 5 years? And what do I think are the most promising? So in the next 5 years, well, we clearly will have the result of the Barnex study. So we know whether that looks like it works or not. We will have the result of my HSN1 study. We will have the result of the SWORD study in the adult reductase inhibitors. And I would be surprised if we don't have a gene silencing trial in CMT1A up and running within 5 years. So that's what I think will happen. But what do I predict is the most promising? Absolutely the generic gene therapies. I think they have revolutionized treating genetic diseases in other genetic diseases, in other inherited diseases. And whether these are the gene silencing or gene editing therapies or the replacing of genes, all of these are very exciting. And the good news is companies are very interested in all of these. And there's multiple new companies developed that are interested in these. And finally, there are challenges to do with CMT. So if you compare CMT to spinal muscular atrophy or Duchenne's, it's over 100 different types versus spinal muscular atrophy. The common type is one type. Most forms are rare. So even though there is a common form, most forms are rare. We really, as a field, have not quite, in my opinion, got it right as how we translate what works in animal models to humans. And that's because I think we need to do the animal trials, which is much rigorousness as we do the human trials. And to date, they probably haven't been. And actually, there's very few people that are expert in both. There are some, but very few. And we have to find a common way of talking together to really make this work. So I'm obviously mainly involved in human studies, and yet I have a lot of work that goes on in the lab on basic science models for genetics. When it comes to trials in animal models, I'm not an expert. So when a company talks to me about something they have done in an animal model, I need people to help me. Was this the right animal? Was this the right outcome measure in the animal? What does this difference mean in an animal? If it runs 2 seconds quicker, is that relevant? We need to really work at outcome measures in slowly progressive diseases. And I would say those of us that are very interested in this need to work with the FDA and EMA to just say we've got to use biomarkers. So I would love to see that we could use something like our MRI protocol for just the first test of these drugs. We don't want patients' time to be wasted and money to be wasted in huge, big trials if we could have found out in much smaller trials early on that it didn't work. So we need to think how to do these trials better. And I think we need to choose the best trial for our patients. And I feel myself very strongly about this. So, patients get tired. Patients are very enthusiastic, but they get tired. So with our natural history study in HSN1, it was an 8-hour visit for patients to do all the outcome measures. So that was tiring. So we need to make sure that if we pick a trial for a patient to be involved in, that we think it's a good trial, we think it's been done well, and we think it will answer the question it's asking. If a trial is done in a way that I don't think is done properly or that it's not using the right outcomes, well, then I won't be involved in that trial, because I think we have to pick the best trials for our patients. And that is an individual decision, both for us and for the patients. So that is, patients sometimes don't like particular kind of trials. I understand that. Patients may not want a trial that means they have to travel to different parts of the country to have testing done in. And in the UK, one of the major problems is, where are the patients? Now, I know this seems a really weird thing to say at the CMTUK annual meeting, and I hope CMTUK will really help with this, because to me, one of the problems is we don't have a good enough national registry in the UK. So what we've done this year, and been very lucky, so I've been trying to fund a national registry of CMT for years, and I had a very generous patient donation last year, which has allowed us to begin to set up the CMT UK registry. And we have other applications in to try to help us fund it. And we want to set up a registry this year, which with patients and clinicians can input data. And we think the power of patients inputting data is it means their clinicians has to, because to be honest, I'm a really busy clinician. If I have to enter 10 people into a database on top of having done my clinic and all my other work, it's really hard to find the time to do it. But if I have a patient hassling me to do it, saying, listen, you've got to do this, I'll do it. So I think it's very important. And it's a very simple registry. We're not trying to collect huge data. We want to know who the patients are, where they are, and would they be interested in clinical trials? So we want to be able to do clinical trials in all of these diseases in the UK because we know where the patients are. I have about 1,700 patients that actively attend me, and about 73% of those have a genetic diagnosis. So I know my group of patients very, very well. And also, we are part of an international consortium, so I know where the patients are around the world. But we need to develop this registry in the UK urgently at the moment. So I'd like to finish by, first of all, very strongly thanking my own group. Many of you will have met some of my group, and all of the work I present is on behalf of the group. Both people who are consultant colleagues recruiting patients, research fellows who do a huge amount of the work, our therapists led by Gita Ramadari, whom I know is very involved with CMTUK, and increasingly our physicists that have let us develop the outcome measures. And I want to thank my current funders, which are long-term funding from the MRC and the NIH, and more recent funding from Wellcome, and also the CMTA and the MDA for funding my more recent studies. And of course, most importantly, I want to thank CMTUK. It's an honour to be a patron of CMTUK. It's a great organisation, and I look forward to it developing further and going forward. And my very final slide is, I just want to really thank all the patients and the controls, that is people who don't have CMT, who have helped us with our natural history studies. None of this can happen without that. I know their hard work coming. I know many of you have been coming to have scans done, since 2009. And it is only now we are actually doing studies using these scans. And I know it's frustrating for patients that things take so long to translate to therapies. But unless we do things properly, every trial we will do will fail. So it really is worth the effort. And I'm incredibly grateful for patients, both for doing these studies and for their patience while we try to develop therapies for patients. Thank you very much.
Thank you so much, Mary, for that. That was a really good update on things, and from understanding a bit better what the Finex trials and where they're at, and the other trials that are going on, the natural history study, which looks to be quite informative and a good way to find out the progression and what's happening in CMT patients. I'm certainly CMTUK very very interested in helping with the patient registry, and we can talk about that. That's something we would definitely want to be involved with in helping you to develop. So we'll talk after this, but yeah, certainly have interest. I've got a number of questions for you. But one question I have, which I hope you can answer. I think we spoke last time at the Q&A about hope, and do you think that we can be more hopeful now? Some of the feedback I got, well, they're always saying we're on the break of something, we're on the verge of something. But right now, do you think we really are on the verge of getting somewhere, if I can put it that way?
No, I'm absolutely aware of how frustrating it is for patients. And I made a mistake, I think it was about 10 years ago, saying to a patient, I expect we'll be in a trial in 5 years. And he turned up 5 years later and said, where's the trial? I never say that now. I'm very, very conservative since then. So I agree how frustrating. And I give talks at the moment called The Cusp of Change, because I genuinely think. So, I'm starting an investigator-led trial. There is another international trial about to start. And I do think the genetic therapies, people might say, well, why hasn't it happened for CMT? It's happened for multiple diseases already. So it is a matter of time and safety. You don't want your child given a therapy at the age of 2 or 3 unless we're absolutely sure that therapy will be safe for life. So I think, there is a safety issue that is much more important, and it will be that we have to try these therapies in adults first. There's no question about that in a disease like CMT. So I do think we're on the cusp of change. As I said, these are 2 rare forms of CMT. The clinical trials are happening now. And I would expect, and I was very conservative and careful in what I said, I would expect a trial of a gene silencing therapy in CMT1A to occur in the next number of years. The other ones are all a little bit more preclinical, and if you ask me to estimate, and I'm involved very heavily in work to try to develop these, it's very difficult to estimate the other forms of CMT. I would be hopeful that some of them have started in the next 5 years, and it may be we can be very lucky like we were with that sword story, very, very lucky that it just overlapped with something else. That's an incredibly, but I can't promise that. And I realize how frustrating it is. It is a little trickier, the gene therapies in CMT, because you've got to get them into nerves. It's not, getting into the brain is easier because you just give a lumbar puncture. It's actually easier to get into nerves all over the body. But there are ways, either by giving it through injections into the blood or giving it through lumbar puncture, that does seem to get into the nerves. And the way that's done has to be optimized. So there's hundreds and thousands of chemists working on the chemistry of optimizing delivery. So I do think we're on the cusp of change. I suppose I would say to patients, I do think we're on the cusp of change. And I would say, if we hand over, it's great when I'm on the thing about on virtual, I can't see anybody giving me a hard look or saying what's you going on about. I would say patient's responsibility is to get a diagnosis. That's the first thing you can do for yourself. And it is easier now. And as I said, you can quote the 78% any clinic you go to, because everyone gets the same panel now. So the first step for treatment is patients having a diagnosis. So there is no patient in the UK with CMT that should not have had that panel done if they want it. That's absolutely through the NHS. And if anyone says you can't, they're wrong.
Right. Well, thank you for that. I think that's comprehensively answered that. I've got some questions here. The first one is from Fran, and she's asking, can you help with which strain of CMT has tremors as a symptom? She has very bad tremors in her left arm.
Yeah, that's a very good question. So multiple forms of CMT. Now, it's much more common to see tremor in demyelinating CMT, that's CMT type 1 and 4, which are both demyelinating, than CMT type 2, but we do see it. And the reason you probably get tremor is thought to be, the tremor usually comes from the brain, what's called the cerebellum, and it's thought to be it's an interruption of the pathway that causes you to be steady because of the neuropathy. And because of the slow conduction in CMT1, we do see more tremor. And it can be very severe, the tremor in CMT. It was actually given its own name. It was called Rosy-Levy syndrome, the type of CMT that had severe tremor years ago. But we mainly see it in CMT1 when it's severe. Very difficult to treat. That's the other question. It is tricky to treat, and it can be very disabling. I appreciate that.
Thank you. And Philip asks, have we derived or do we anticipate useful results from the Accelerating Clinical Trials study?
Yes. So that's a very important one. And maybe I'd say that's Accelerated Clinical Trial Study is a trial. It's a multi-center study in 5 centers funded by the NIH. And it's led by the Rochester Group. And we're the UK center. There's a Italian center and 3 centers in the US. And this is a study. We've done the work in CMT1A to show that MRI is a good outcome measure. But this looks at MRI, which we're doing as part of it, versus all of the other clinical trial measures that are out there. And it has developed a new outcome measure called the CMT form, which many of you that come to my clinic and my research will have seen, where it's a way of actually looking at the function of a patient. Can you walk quickly? Can you do the 6 minutes? Can you get up and down from a chair? So it's a new outcome measure for CMT, and it's comparing it against the other ones. We're halfway through. We're fully recruited. And I have to say to patients again, we are fully recruited, including in the UK, despite COVID, because patients came. Now, we obviously put huge safety measures in place and provided transport, but we are incredibly grateful. So that will deliver. And we think what that will be important for is when we go to the FDA and EMA to say, can a clinical trial happen, use these outcome measures? So at the end of this study, we hope to have a package of outcome measures, which are the gold standard to do trials. So it will definitely 100% deliver. And in my own study of MDA, looking at CMT 1X, 1B, and 2A, we've shown exactly the same thing with the MRI. So I think even though the ACT Accelerate trials is only in CMT 1A, my other parallel studies will also show it in 1X, 1B. And I think if you show it in 4 or 5 types of CMT, it will work for all the other types, as long as we're talking about the types that start in the legs, so that the length-dependent ones. The rare forms of CMT that start in the arms will need different outcome measures.
Thank you. And I have a question now from Nikki, who asks whether people who have a diagnosis, should they get retested using WGS?
No, if you have a diagnosis, the old methods were extremely accurate. It's just that they were slow. So if I now test 100 genes, I can test a panel of 100 genes and get the results in a few months. If I did those 100 genes in the past, it would take me a year per gene. So it's actually interesting. The diagnostic rate before we had the whole genome was about 67%. It was still really good. So it's not that we're doing a huge amount more diagnosis. We're just able to do it much, much faster. So if you have a robust diagnosis, it won't be wrong. It's very unusual. The only thing I'd say, and this is a very small percentage of patients. So when I used to do diagnosis in the past, I would do one gene. If it was negative, I'd do the next gene. But say I found it that the third gene was abnormal, I would stop. And we now know that about 2% of patients with CMT have probably got two genes causing it. So we now would pick that up because we do them all together. So if patients come back to me and they're unusual, so if they've got a form of CMT and we know the gene, but they're not usual, I will then test again to see if there's any other genes. But in the vast majority of patients, the way we used to test was absolutely fine. It was just slow.
Right, thank you. Caroline asks, her daughter has recently, I think she means diagnosed with CMT2 with a de novo mutation on the MORC2 gene. Essentially, what she's asking is, are there any trials taking place that would look at this type of CMT?
So this is a very rare type of CMT that's been relatively recently, the gene has been found within the last 5 or 6 years, and I have patients with it, and it is at a much earlier stage of development. It's a complex gene. It's not an easy gene to study. So yes, there is work, but the work hasn't identified a candidate therapy yet. So it's at the stage still of working out how the gene causes the disease. So it's not at the stage where it is even got a candidate therapy. So for those rare forms, that's one form of research that needs to happen. But the other one is pathway therapies. So if there was something that could just treat nerves, but they're again slightly further off. So MORC2 is one of the ones that's pretty rare and is pretty far back in the development.
Thank you. And Ramona is asking, how do people get involved in trials?
So I think that's a very good question. So traditionally in trials, I mean, I run a very big group, so I usually recruit for trials through my own patients. And, we'll see any patient in the UK that wants to come and see us. And we often see patients as a one-off visit so that we see them, register them and have them on our database forever. And then they go back to their local neurologist, their local centre. And that's been very helpful. But going forward, the reason we really want to set up a good registry is that, we would run a registry, and hopefully with the help of CMTUK and other people, that you could register to be interested in trial and just be on the registry. And when there's a trial available, we will contact you. So that's what we really, really need in the UK. We need to know who's interested. Where are you and if you're interested? And that's what we will be setting up this year. But that's what we're already in the process. We won't be setting up. We're already well in the process of it.
I have some patients here saying that they want to go on the registry, so that's good.
It's good. And I'm hoping to the CMTUK as soon as we are live, because my usual naivety, I thought we could set this up really quickly. And there's all kinds of things you have to do, permissions to get to have patients allowing you to put their data in a registry and to have the right registry. But we're much closer to it than we were. And as I said, we have had some seed funding, which is excellent.
Brilliant. Good. Alison asks if her CMT has not yet been diagnosed despite genetic testing. How likely is it that a diagnosis will be made in the future?
100%. So I can easily say that, can't I? Because it's a future question. But 78% of my 78% of my patients are now diagnosed. I mean, that was 0% in 1990. So we will find all the genes. I will not tell you when, because some of the rare ones we have found, the mechanism underlying their CMT with whole genome sequencing is incredibly complex. We have published a gene a few years ago, and the amount was a decade of work to find it. So I think they will be found. The most important thing, if you want to find your type of CMT, is to be enrolled in a research study, which would find it. And the great thing about being enrolled in a research study for CMT and whole genome. So one thing issues about the 100,000 genome, it's great. But like people that are entered into it, if they're entered into it, when I see them, with their permission, I can access their data to look on the research side. But if you had your CMT diagnosis done in Southampton and wanted me to look at your whole genome, It's incredibly difficult for me to get permission to do, so I can do, but incredibly difficult. So in the UK, we need to be better at sharing data. So if you have whole genome sequencing done, the great thing about it now with the new platform, so everyone that gets a diagnosis of CMT will have it done, except 1A. 1A is easy to diagnose in a different way. You just get it done with a simple test. But everyone else will have their whole genome sequenced to look for the genes. And then if you're negative, it's a bit like having it in the bank. It is there. And every time a new gene is found, people can look at your data to see if it's there because you've been sequenced. We just don't know where the gene is. So to me, it's like putting it in the bank so that eventually it will be looked at. What the UK hasn't quite got right is giving people access to the bank if they want it. So I think you should be able to tell me I can look at your DNA because you've given permission. But it's not as easy as that. It's still some clunkiness in the system. But I hope with the new consent forms that say you consent for research on your gene as well as diagnostic, that will be possible. Because at the moment, if you consent for genetic testing, you're only consenting for us to look for what's known today, not for what will be known in the future.
Yeah, so it's a usual thing, really, being very secure with the information, but making sure the right people can get access to it.
Yeah, I mean, I think it's the balance, isn't it? I mean, I know there's a lot of talk about this in other forms of life. It's the balance between data protection and actually you having the right. And in the end of the day, as far as I'm concerned, as patients have the ultimate right, a complete right. They're the only ones that can say what their data is ever used for. And they're the only ones that can say, but they have the right to say it can be looked at as well as it cannot be looked at. I often feel that the default is always the cannot do, and that I have to work much harder to be allowed to do things. So I think we need to just make sure that patients feel that they are having access to the kind of research they want to be involved in without barriers.
Yeah. Okay. Got 2 or 3 more if you're okay for time. Jeff is asking, is CMT progressive within hands as much or more than in feet?
So I think that's an incredibly good question. So CMT generally, most forms, is length dependent. So the longest nerves in your body are to your feet. So it often starts in the feet. And then, the arms are pretty long as well. So not long after the feet, the arms become involved. Some people say a few years, some people say 10 years, some forms are quicker. But once the arms become involved at the hands, they will progress at the same rate as the feet. So just because it started later, it'll still progress. Some forms of CMT are more severe. And I would say I notice with patients, understandably. So you use your legs, of course, a lot, don't you? You use your legs to walk and do everything. But when your legs don't work properly from when you're younger, you get used to it. And you modulate your life to accept that. You might wear splints, you might use a stake, you might, you're not going to go and do 100 meter, you're not going to go and be a fast 100 meter runner. You modulate your life. But your hands are incredibly important for everything. So hand function, I think, it's that you notice tiny differences more. So if your big toe is weak and your second toe becomes a bit weak, you hardly ever notice it. But if your fingers get a tiny bit weaker, you notice it in real life because we use our hands all the time. So I think it's that little changes in hands are much more eloquent in terms of causing problems. So I think patients often find in their middle years that they really begin to notice their hand function is a problem. Now, you could say life has improved, that we don't use our hands for as much as we used to for fine things like writing, and that computers have been adapted so that we can, there's all kinds of adaptations and voice activation, etc. So life isn't as dependent on hands. But some people love doing needlework or doing sports that involve your hands or playing the piano or doing stuff, painting, that the hands are critical for. So I think it is difficult because the hands, and if you have a severe tremor, it is difficult as well. So it's just as progressive in the hands as in the legs. It's just that it starts later in most forms.
Yeah, I think that makes complete sense, actually. That's thank you. And Therese has concerns around swelling around her feet and ankles and lower legs. There's no actual question there, but she has some concerns about that.
So I think that's an incredibly important comment to make. I mean, I think, we don't know why some patients have such problem with swelling and others don't. So there's no doubt with any neuropathy, the problem with neuropathy is your muscles are not moving. They're not moving normally. And that does mean the muscles around blood vessels aren't moving normally as well. So it does mean you get what's called edema. That is swelling of your legs because your muscles are not pumping the blood around properly and you get an accumulation. And that can cause discoloration. Some people get severe discoloration, but the edema can be very severe in some patients, and it's often better after sleeping overnight when your legs have been up and made worse during the day. And there are some very good clinics to manage edema, mainly with, you know, various kind of special kind of stockings, etc. Sometimes with other things that are needed, sometimes with medication. But I think it can be a very severe problem. But most parts of the country do have good edema managing clinics because of other diseases that causes, not because of CMT. But I think it can be a very troublesome symptom. And an overlooked symptom. It's one of those things that doesn't get talked about as much as it should do.
Yeah. And Nikki has another question here about, is there a risk of having too little PMP22 if you turn off the gene?
Of course, I hope I made that clear. You're absolutely right. If you've only 50% of PMP22, you have another neuropathy called hereditary neuropathy with liability to pressure palsy. So we absolutely have to not turn off too much. Now, to date in the studies done, it does tend to turn off about 30%, which is about what we want. And it looks like that's perfectly safe. But, you know, we have to look at this in humans. So I think the safety studies, when we get the gene silencing therapies, the early safety studies to actually measure the PMP22 expression. I think the really good thing is that used to be very difficult to measure. And John Savern, a US scientist, has really developed a very reliable way of measuring it by doing skin biopsies. Because we really need to measure properly that we're not overcooking it, if you want to put it that way. Very, very important question, and something we're very, very aware of. And, you know, for some reason, we don't really understand the dose of PMP22 is critical to nerves. We know that because there's diseases with too much and too little. To be fair, we don't quite understand why it is so critical.
Okay. And Nikki's other question is, there is such a large crossover of proteins, genes involved. Is it possible for someone to develop more mutations throughout life?
A very good question. The answer is no in this case. You do see it in mitochondrial disease, which is a different kind of genetics, but in these diseases, you don't acquire mutations in your DNA. And I think I'll just explain that a bit more. The basic DNA in our nucleus in our cells is set. You don't change that. You can develop, when people develop tumors, they develop mutations in the tissue, which is a tumor, but that's different. So in diseases like CMT, if you have a mutation, it's there from birth, it's there for life, and it doesn't change, and you won't develop new ones.
Right, that's answered that. Yes, thank you. That comes up. And then I have a question from the office, which comes up quite a lot. Well, not frequently, but not infrequently. Sex drive and incontinence. I don't know if you can
Sure.
say a bit about those two.
Yeah, so most patients with CMT, so the peripheral nervous system, which CMT mainly affects, is the motor and sensory nerves. So if you want to move your hand or your leg, it's the motor nerves from the spinal cord to the hands and the legs. The sensory nerves are the ones that go from the skin back up the spinal cord. So that most forms of CMT affect those. It is extremely rare for CMT to affect what's called the autonomic nervous system. So the autonomic nervous system is the part of the nervous system that does all the automatic stuff. So it controls your blood pressure, controls your pulse rate, controls urinary function, bowel function, sexual function. So very, very rare forms of CMT can. These are like, they wouldn't have even been called CMT. They're called, some are called hereditary sensory and autonomic neuropathy, but there is an overlap with some of those. So I would say most forms of CMT don't. But if you have very, very severe sensory involvement as your CMT, you might lose the sensation of passing water, or you might lose the sensation of passing a bowel motion just because the sensation is so bad, but they're really rare forms. And sometimes if you lose the sensation of passing water, you might get a big bladder. You might not really know when you want to go to the loo. And similarly with sexual function, people could lose sensation. Now, in my experience, it's extremely rare in most forms of CMT to affect these functions. But there are rare forms which it can do. But I would say if anyone has those problems and your GP or doctors are telling you it's due to CMT, just query that, because unless it's a form of CMT that does affect that, it's usually more likely to be something else.
Yeah, that raises a very good point, because often people get there's a misconception maybe with the medical profession, maybe with patients that all the symptoms they've got often get referred to CMT, and of course, just having CMT doesn't mean you won't get other things. So,
absolutely, I saw a patient in clinic recently who, you know, had CMT, genetically proven, and the fact is 90% of their symptoms were due to another condition which hadn't been picked up. So I think you're absolutely right. You know, most GPs will only have 0 to 2 or 3 people with CMT, you know, in their practice. They'll either have a whole family or they'll have nobody. That's, you know, to think about the incidence of it, it's about 1 in 2,500. So, you know, they're not going to be experts in it. And you can't expect a GP to be an expert. They might look it up. Luckily, nowadays, you can look things up really quickly. If you're about to see it, you know, you can go online and look it up. But you're going to be more of an expert as a patient. So I think, you know, you absolutely, if you develop problems, and I think it's especially if people develop joint problems, you know, if they develop a frozen shoulder, for instance, or other problems, and maybe the CMT made them more at risk of those problems because they don't move as well as other people. But that's not due to their CMT. CMT needs to be treated separately. So really important that patients are, you know, that they are really listened to make sure it's not all assumed. And I agree with you, that's very common, that misconception.
Yeah. And one final question is from Andrew, whose podiatrist advises him not to use insoles or orthotics yet. At what point should he start using them? Is there a risk from starting too early?
Yeah. So I wouldn't comment on an individual person that I haven't seen because it's absolutely individual. I think it's, you know, there are some people who hate orthotics, who basically, we think they should be wearing them. They wear them, they walk better, but they hate them. They feel that they're just, you know, they don't like wearing them. They feel they walk better without them. So it's very individual. When is too early? Well, you know, sometimes people say they don't like to wear them because they think if they wear an ankle foot orthosis, they won't use their muscles as much and maybe they lose some function. I think that, you know, there's some truth to that if you have some good muscles. So I think you use them when you need them. So if somebody is falling or tripping all the time and they walk much better with the orthosis, you wear them. There's no reason not to wear them. With insoles, it's often for things like pain. It's often for discomfort when you walk. And, you know, people without CMT wear insoles to make things more comfortable. So it's a completely individualized. Now, if you have a good podiatrists are much more expert in feet than any doctor. Good podiatrists, good chiropodists, they know feet. And they will be often the right people to advise you. And but I don't think there's a right or wrong answer. It depends on your type of CMT, and it depends on you and what you do. You know, if your job involves you walking a lot versus sitting in an office all day, you have different requirements. And I have many patients who wear different orthoses or insoles for one part of their life or for the other part of their life. So I think it's a very individual, and there's no right and wrong answer to that.
Brilliant. Well, that's all the questions. I don't think I've missed any, but there we go. Thank you so much, Mary, for doing that and for giving us your time this morning. That's been fantastic.
Okay, thank you very much.
Take care now. Thank you. Bye-bye.